So here we are in the last part of the lab, and I just want to show you a few things that I use in the reactions that I'm running when I'm trying to repair damaged DNA. So I've got some water here, so I'm just going to show you the vortex. So this is our vortex. I just take my sample and I press down, and that will mix it. You can press anywhere, so you can press in the middle or on the side, and sometimes we lie it down so it spins like that, or hold it up like that too. So that's a vortex, and we'll always vortex our samples to mix everything together. We'll also use this centrifuge. So when I press the lid down, my sample will spin around, and that will also help everything to mix together. We can see two different attachments here. So this is the large attachment, which is this sample size, and there's also a smaller attachment for smaller PCR tubes too. We also have the PCR here, which I use in my reactions to incubate my samples at 37 degrees for an hour. You can change the temperature of this, but we always use 37 degrees because that simulates body temperature. So we want to simulate body temperature because that is going to say if this enzyme was in a human, how would we repair the DNA? How well will it repair DNA in humans? So that's why we always want to be able to have that data to compare to. So when I'm doing my reactions, I know that if this was in a human, I would get this amount percentage of repair. So that's when it ties in with the HPLC, so I can analyze my data. And then finally, we have this section, which is the protein purification preparation. We can see right there, there's a sample of ABH2 being purified. So this is where graduate students in our lab are purifying the proteins for us to use on our data analysis. So that concludes the tour of Dr. Lee's lab. I hope you enjoyed joining me on it, and thank you very much.